Morning everyone. My topic is media for fermentation, and it includes the typical media, composition of the media, formulation, carbon, nitrogen, minerals, and energy sources. Fermentation media. The fermentation media must be good, adequate, and industrially usable medium, and it must be capable to bring out the industrial potential. The major cost determining factor is the fermentation media, and it account up for the 60 to 80% of the process expenditure. And the media is very based on the scale of the production, that is a laboratory scale, pilot scale, and industrial scale, and the end product biomass, primary or secondary metabolites. Two types of industrial media are used, that is the seed fermenter and the protection fermenter. And the liquid media are more generally used than the solid media because it requires less space, it is more amenable to engineering process, and it eliminates the cost of providing agar and other solid agents. The industrial medium is usually made with unpurified raw materials, that is derived from the natural plants and animal extracts and byproducts of the other industries. Pure chemicals are not usually used because of the high expense. Industrial media, it must satisfy the needs of the organisms, and the media must contain carbon sources, nitrogen sources, mineral sources, growth factors, and water. The proportion of each components will vary depending upon the type of the fermentation. It must not contain materials which are inhibitory to growth, and media formulation can be done by analyzing element composition of the industrial microorganism. And the criteria for the industrial media includes the cost of the material, and the cheaper raw materials must be selected, and the raw materials usually includes the waste products from the industries like corn steep liquor and molasses, and ready available of the raw material must be easily available and fulfill the technical objectives of the process, and there must need the uniformity in the quality of the raw material in ease of the standardization. Transportation cost must be very low, and it must support the optimum biomass. Concentration of the product, its rate of the formation and yield of the substrate must be utilized. Adequate chemical composition of the media must be provided. Suitability with the fermentation process, and the presence of the relevant precursors like cobalt is used as a precursor in the vitamin B12 biofermentation process, and the fermenter must contain easy aeration, agitation, extraction, and purification, along with formulation, mixing, complexing, viscosity characteristics. Sterilization requirements at any potential denaturation problems must be included. Minimum undesired products must be produced at the end. Ease of the disposal of the wastes resulting from the raw materials. Overall health and safety implications. And the media, basic formulation of the media must include, and the basic components for the media must include the biomass production, metabolite production, energy, and components of the cell maintenance. And it must include carbon and energy sources, nitrogen sources, oxygen, and other requirements for the production of the biomass, other products, carbon dioxide, water, and heat. And the carbon sources, first one is the carbon sources that is necessary in the medium, and it must be essential for the biomass and product formation, and must have a good impact on production cost. Strategic planning is required to ensure the constant availability. Cost of the carbon source is inversely proportional to the purity and the quality. Ease of sterilization and gelatinization properties are also important. A government legislation may prevent the use of some carbon sources. And the carbon requirements is necessary for the cells for maintaining to the requirement of the growth, cell maintenance, product formation, provide energy. The biomass yield coefficient, it is also called as the cellular yield coefficient, and it is the weight of the organism produced from the given weight of the carbohydrates. By utilizing the carbohydrates, the given weight produced is known as the biomass yield coefficient. It is Y carbon. It is detected as the Y carbon. It is the biomass produced by the carbon substrate utilized. And carbon source types, it includes the carbohydrates, hydrocarbons, alcohols, alkanes, organic acids, animal fats and plant oils, hydrocarbons or methanol, and carbon dioxide for the photodrops. Pure glucose and sucrose are rarely used for industrial scale fermentations and rarely for metabolized sugars. Next one is the molasses, and it is the byproduct of the cane and beet sugar production. It is cheaper and more useful source of the sucrose, and molasses are residues remaining after the most of the sucrose has been crystallized from the plant extract. And it is a dark colored viscous syrup that contains 50 to 60% of the carbohydrates, that is primarily sucrose, 2% of the nitrogenous substances, and some vitamins and minerals. And the overall composition, it varies, it depending upon the plant source, location of the crop, climatic condition under which it is grown, type of the factory where it is produced, and it is used as a substrate for ethanol, organic acid, SCP, and also for enzymes. And carbohydrate concentration may be reduced during the storage by contaminating microorganisms. Hydrol molasses, it is also a type of the molasses. It is a byproduct of maize starch processing, primarily contain glucose. Next one is malt extract. It is the aqueous extract of malted barley is concentrated to form malt syrup. It is a good carbon sources for the cultivation of the filamentous fungi, yeast, and actinomycetes. And the composition, it consists of the 90% of carbohydrate, 5% of the nitrogenous substances, proteins, peptides, and amino acids, etc. Starch and dextrins. It is not readily used in the fermentation process, and it is used for the production of filamentous fungi that produces amylases and can be used these. Maize starch is most commonly used, and starch can be converted to the sugar syrup. Glutamic acid production using hydrolyzed cassava starch is an example. The next one is the sulfide waste liquor, and it is derived from the paper pulping industry, primarily used for the yeast cultivation. And liquors from the coniferous trees contains 2 to 3% of this sugar, that is hexose is 80% and pentose is 20%. And liquors from deciduous trees has mainly pentoses. And the liquid is processed to remove the sulfur dioxide by the precipitation or stream stripping. Low pH is adjusted with calcium hydroxide and supplemented with nitrogen. Next one is the cellulose, and it is predominantly found in the lignocellulose in the plant cell walls. It is mainly found in the plant cell walls, and it is valuable renewable source for the bioconversion to ethanol for fuel use. And lignocellulose is composed of cellulose, hemicellulose, lignin, etc. And it is available from the agricultural, forestry, industrial, and domestic wastes, etc. Only a few microorganisms can utilize this lignocellulose directly and for the use for the fermentation process, and pre-treatment is required for this, and mainly used in the solid substrate fermentation. Another example is the wood molasses and rice straw. Whey, it is an aqueous byproduct of the dairy industry. The annual worldwide production is 80 million tons, and it is expensive to store and transport. And lactose concentrates. This lactose concentrates is the evaporation of the whey and removal of the milk protein for use as food supplement. Lactose is metabolized only by fewer microorganisms, and it is extensively used in the penicillin fermentations. Next that must be produced is alkanes. Alkanes is the mixture of N-alkanes are used, and cost is depending on prevailing price of petroleum. The main example for alkane is methanol. It is more preferred than methane. It is high purity methanol is readily obtained, and it is completely miscible with water, and the high percentage of carbon and energy content is present in there, that is necessary for the fermentation process. It is relatively cheap. Higher concentration are toxic, hence low concentration must be produced, and oxygen demand and the heat of the fermentation are high. It can be used for the SCP production and ECA effluent treatment and downstream processing. Next one is alcohol. The important alcohol that must be provided is ethanol. It is less toxic than methanol and used as a salt for co-substrate by many organisms. Too expensive for general use as a carbon source, and biotransformation to acetic acid is a major fermentation process. Next one is the fats and oils. It is 2.4 times energy than carbohydrates, and it occupies less volume. It is useful in fed batch operations, and we must produce the antiform activity, and hard animal fats are rarely used in the fermentation. Most commonly used fats and oils are plant oils and fish oils, which is a primary and supplementary carbon sources used in the antibiotic production. The plant oil that commonly used, it includes cottonseed, maize, olive, palm, rape seeds and soya, and it is composed of oleic, linoleic and linolenic acids. And next one is the nitrogen sources. These type in earlier we discussed the type of the carbon sources. Now we are going to discuss about the nitrogen sources. And nitrogen, it is used for the growth of the organisms. It can be classified into inorganic and organic type. And inorganic type of the nitrogen sources, it includes the ammonia gas, ammonium salt, nitrate, etc. And organic sources of the nitrogen, it includes amino acids, proteins, urea, corn seed liquor, soya meal, peanut meal, soybean hydrolysate has methionine and threonine in addition to the organic nitrogen, which is also helpful as an additive in the fermentation process. Most cells use ammonia or other nitrogen source, and preferred nitrogen source is ammonia in the form of reductase. And ammonia can be used to adjust the pH. Rapidly utilizable nitrogen source, it inhibits the antibiotic production, and nitrogen is often supplemented in the crude forms. Next type of the nitrogen source is the corn seed liquor. It is the byproduct of the starch extraction from maize, and first use of it is in the penicillin production fermentation process, and its composition, it is made up of the, depending upon the quality of the maize processing condition, it must contain 4% of the nitrogen, wide range of the amino acids, vitamins and minerals, residual sugars, and it can be replaced by similar liquids such as those derived from potato starch protection. Next one is yeast extract, and produced from waste bakers and brewers yeast, and also use candidas utilis cultivated on the waste from the wood and paper processing and fermentation media are salt free, and the yeast cell hydrolysis process is called as the autolysis, and it is initiated by the temperature or osmotic shock. Next we are going to discuss about the autolysis. It is initiated by the temperature or the osmotic shock. It do not activate the yeast enzymes 50 to 55 degrees Celsius for several hours, then raised to 75 degrees to inactivate the enzymes, and cells disruptions, that is a plasmolysis or mechanical disruption. Cell wall materials are removed by filtration or centrifugation. Resultant extract is rapidly concentrated, containing 50 to 60% of solids. Next one is the peptones. Too expensive for the large-scale industrial fermentation. It is prepared by the acid or enzyme hydrolysis of high-protein materials, that is meat, casein, keratin, peanut, soy meal, and their amino acid composition vary depending upon the original protein source. Keratin peptone is rich in proline and cysteine but lacks lysine, and peptones from plant sources contains really large quantities of carbohydrates along with the nitrogen. Soy meal and the residues remaining after extraction oil from soya beans, and the composition of 50% of protein, 8% of the nitrogenous compounds, and 30% of carbohydrates, and 1% of oil. Next important component for the fermentation process media is the water. Water is used for the heating, cooling, rinsing, cleaning and fermentation, and large quantities of clean water must be provided, and we often choose for the tap water, that is clearly cheaper, and soft water is highly expensive, so that is not commonly used. And determination of the water content must be frequently checked, like the mineral content analysis, and the mineral content that affect the fermentation, pH, dissolved salt and effluent contamination must be frequently checked, and water may also provide trace mineral elements. Before use, the removal of the suspended solid colloids and microorganisms must be removed, and hard water is treated to remove the salts such as calcium carbonate, iron, and chlorine may also require removal. Salt addition may be required, and plant and animal cell culture highly purified water is required, and recyclable or reusage of the water is commonly used. And the another type of the components needed for the fermentation media is the minerals, and the main functions of the minerals is to provide osmotic balance of the media, developing buffer system, cofactor for an enzymes, and the special requirements is the essential and the non-essential type of the minerals that must be provided. And the essential minerals includes the Mg, potassium, sulfur, calcium, chlorine, and copper, cobalt, iron, magnesium, molybdenum, and zinc get from impurities. They act as the non-essential minerals, and chelators must be also be provided for the fermentation process. And these main sources of these type of minerals is the yeast extract or the peptones. Vitamins and growth factors must be provided, and vitamins, it includes the different type of the vitamins, amino acids, nucleotides, fatty acids, and sterols, and many bacteria can synthesize all necessary type of the vitamins. Filamentous fungi and yeast supplement the medium. Most natural carbon and nitrogen also contains required vitamins. Next is the buffer, calcium carbonate, that is of the pH 7. Carbon and nitrogen sources, that is amino acid, peptides, and protein. pH can be controlled by acid and alkali additions. Metabolic regulators must be provided, that is precursors, inhibitors, and inducers. And precursors, it is the substances that enhances the reaction in the fermentation process. Some fermentation needs specific precursors to increases the product yield, and often added in controlled quantities and relatively in pure form. Phenylacetic acid is added as a side chain precursors in the penicillin production, and D3-threonine, it is used as in the precursor in L-isoleucine production by Serratia marcescens. Inducers and elicitators, specific inducer compounds or a structural analog may induce product formation, incorporated into the culture medium or added at a specific point during the fermentation. And production of flavonoids is triggered by pinostrobin and necessary in fermentation of the genetically modified of the microorganisms. Starch for amylase production. And inhibitors, it includes redirect metabolism towards the target product. It reduces formation of the undesired intermediates. It halts a pathway to prevent further metabolism of the target product. Sodium bisulfate, that acts as an inhibitor for the production of the glycerol. And GMMs is antibiotics in medium used to selectively inhibit the plasmid free cells. Bromine inhibit chloride tetracycline in the tetracycline production. Next one is the cell permeability modifiers. It modifies the cell walls or membranes that promote the release of the intracellular products in the fermentation medium. Example is the penicillin and surfactants used in the amino acid fermentation. Oxygen must be provided for the medium. Dissolved oxygen levels are important, control the growth rate and the metabolite production. Air containing about 21% of oxygen or occasionally as pure oxygen can be used. Oxygen requirements vary widely depending upon the carbon source. For most fermentation, the air or oxygen supply is filtered, sterilized prior to be injected into the fermenter. Antiforms must be added. It is used to prevent the forming. Forming is largely due to the media proteins, and proteins become attached to the air-broth interface. It denatures to form the stable form skin at the top, and it traps the cells, autolysis, and it can cause and it prevent the for the forming. And forming can cause many detrimental effects on the fermentation process. Hence anti-forming is necessary because it causes the detrimental effects like reducing working volume of the medium, decreased mass and heat transfer, blocked air filters, loss of the aseptic conditions, loss of the productivity, need to allow freeboard, etc.